Hop on a plane to Florida, visit the dentist for your annual checkup, or simply take a walk down the street. These seemingly unrelated actions all have something in common. It's called radiation. Every second now, 5 cosmic particles are hitting my hand. We're constantly exposed to radiation, either from natural sources, such as sunlight or cosmic rays, or through life-saving medical procedures, including X-rays or cancer radiation therapy. Doctors seeking to destroy cancerous cells beam them with radiation. This damages the cell's DNA, killing the tumor. But if used incorrectly, it also damages healthy cells and causes side effects. I'm walking down a shaft at the heart of the Weizmann Institute's particle accelerator, where a team of physicists, led by Professor Amos Breskin, have come up with a surprising twist to tackle this key medical challenge.
Hi, come and join me.
So it would seem then that this radiation is a bit of a double-edged sword.
Well, this is true in part, particularly when you want to treat cancer, to kill cancerous cells. In order to do it, we have to use radiation. We have to damage the DNA molecules which constitute the cells. And here we have a problem. You know that the DNA has a double-strand structure. If we irradiate this molecule with a dose which is too low, we will break only one of the strands of the DNA, and therefore the DNA will be able to repair itself, and the cancer will continue and proliferate. On the other hand, if the dose is very high, then we have a high chance to kill the cancer, but also normal cells will get hit and damaged, and this is not what we want to do.
Is it possible to determine the exact dose of radiation needed?
Well, that's precisely what we are doing. Come join me, I will show you the experiment. So here we are crossing the experimental hall of the accelerator, which is a giant machine, generally used by physicists to explore the basic structure of matter. What we are doing here, we are using the same machine to explore the effects of radiation on the living cell. Remember the DNA molecule? We are simulating it by a very tiny gas bubble which serves as a model of the DNA molecule. Now we have our particles coming all along this tube from the accelerator. They are heating the gas bubble, they are depositing some energy, ionizing it, and releasing electrons and ions. Now by very sensitive detectors, we have to count, for example, the ions which are produced by each particle, and knowing the number of the ions and the location of their deposition, we can know something about the possible effects of these ions on the real DNA molecule.
So this is what we're seeing, in fact?
Precisely. What you see here is a single event deposited by one proton. You see that each of these pulses represents a single ion. For example, an event in which you have one ion is absolutely non-harmful. Well, an event like that in which there are many ions deposited in a very small space are fatal events which may destroy totally the DNA molecule, and these are the events we are searching for.
This system, currently being tested in California's Loma Linda University Medical Center, is far more precise than existing technologies. In the future, it may enable clinicians to adjust radiation levels to the needs of individual patients, a feature that will surely save many lives. On other fronts, the new application may help set safety standards for exposure from other radiation sources, such as those encountered in the workplace or during routine X-rays.